Hi everyone, welcome to the Headwall Photonics Harmful Algal Bloom UAV Study webinar. We're just going to give a couple minutes for folks who are arriving a little bit late or having trouble getting on. So we'll start in about 5 minutes. Just going to wait a couple more minutes and then we'll get started. Thank you for coming.
Those are pretty good. You want to start?
Okay. All right, everyone, we're going to get started. Thank you again for attending the Headwall Photonics Harmful Algal Bloom UAV Study webinar. This webinar is being recorded, excuse me, and should be available for viewing within 48 hours. Excuse me, that's quite the frog in my throat. I am Marissa DuPont, Marketing Specialist here at Headwall. If you have any questions, please use the questions panel in your GoToWebinar control panel. We will answer as many of your questions live as possible, but additional questions will be answered by email after the conclusion of this webinar, and you can send those to marketing@headwallphotonics.com. We'd like to welcome our panelists, Mikhail Jobran, who is the Regional Sales Manager of Headwall Photonics, US Northeast, John Margison, who is the Product Manager at Headwall Photonics and FAA Certified Drone Pilot, Carson Roberts, Ph.D., who is the Principal Applications Engineer here, and also an FAA Certified Drone Pilot, and Scott Gallager, who is the President of Coastal Ocean Vision and former scientist at Woods Hole Oceanographic. Now I'm going to turn it over to Mikhail, who will continue our presentation.
Hello, everyone, and welcome to today's webinar. My name is Mikhail Jobran. I'm the Northeast Sales Manager at Headwall Photonics. At the beginning of this webinar, and before we dig deeper into today's topic, I want to give you a quick introduction to Headwall Photonics. Headwall Photonics have been in business since 2003 and was recently acquired by Arsenal Capital Partners. We have 3 locations in Massachusetts, including our headquarters in Bolton, and a site in the Netherlands. Headwall Photonics is ISO 9001 certified with 40,000 square feet of engineering and manufacturing facilities. Next slide, please. Headwall Photonics is a leading supplier of hyperspectral imaging solutions used for machine vision and remote sensing applications. Headwall also specializes in manufacturing optical components and assemblies such as diffraction gratings, spectrometers, and spectrographs, which we normally sell to OEMs and end users. Next slide, please. In this slide, I wanted to show you some examples of our fully integrated turnkey airborne packages. All our turnkey airborne packages include a UAV, hyperspectral payload, high-performance GPS IMU, software, training, Pelican cases, and all the accessories needed. You also can get it with a 16-channel LiDAR, which is optional, depending on the applications that you're working on. Payloads could be a VNIR payload, a SWIR, or both. All of our turnkey packages are ready to use out of the box. Next slide, please. In this slide, you can see a list of our key remote sensing applications for Earth monitoring, research and academia, government, and commercial remote sensing. Most of these applications were done with our off-the-shelf sensors and turnkey airborne packages. Please do not hesitate to contact us if you have any questions regarding our products and applications. Thank you, everyone, and now I will hand this over to John. Go ahead, John.
Okay, thanks, Mikhail. As mentioned, I'm John Margeson, Product Manager for Remote Sensing Products here at Headwall. For those who may not be familiar, I'm just going to give a quick intro to the basics of hyperspectral imaging and how it's used in remote sensing applications. So to put it simply, hyperspectral imaging is a powerful measurement technique which combines the spatial information of an image with the chemical discrimination of spectroscopy. So you can start to conceptualize this by first thinking of a typical camera like you may have on your phone. So these are going to work by capturing a snapshot of visible light 3 times in 3 specific bands of colors, typically a red, green, and a blue, which you can think of as an RGB camera. So those 3 subsets are then overlaid so each pixel in the image contains a specific blend of that red, green, and blue to simulate other various colors, with the end result being an image which closely represents what the human eye can see. So if we compare this to hyperspectral imaging, which can capture the same image but across hundreds of bands of lights, spanning both the visible range and beyond, we can create what was called a data cube. So essentially a data cube is a stack of the same image measured hundreds of times across each specific wavelength of light. So these data cubes are very data intensive and they require specific software to analyze, but they provide us with continuous spectral information for each pixel within an image. And this can be very useful because, you can go to the next slide, please. Because with continuous spectral information, we're actually given the ability to differentiate between objects which might appear similar to our naked eye. As it turns out, all objects, organic and inorganic, have a fairly unique curve when we measure their reflectance across numerous wavelengths of light. This is essentially their spectral fingerprint, or we would call their spectral signature. And once we've identified a spectral signature for an object, we can use it to analyze our hyperspectral data cube for that image and define the chemical composition of the objects and materials in that image. So we can refer to this on the right-hand side of the screen as compared to multispectral cameras, which also provide some level of precision into the spectral signature beyond the visible light. However, a multispectral camera is going to lack the number of data points which hyperspectral imaging can provide. Therefore, they aren't nearly as precise in their identification. Go next slide. Now, it's important to point out not just how hyperspectral data works, but how hyperspectral data is captured in remote sensing applications. So, for example, here I have 3 representations of the same image. In this case, a nice coastline. Presumably, this image was taken from a UAV or a small airplane. On the left-hand side, you'll see the full 2D view of this image. In the middle view, you'll see how a typical snapshot camera would capture this image in a 2D array, so in two dimensions. However, you'll notice on the right-hand side, this image is captured with our hyperspectral sensors. This is in what is done, it's called a push broom technique. In this technique, an image is captured across 1 row of pixels at a time, relying on the motion of the sensor to capture the next row. These lines of pixels are then stitched together to create our data cube. That means that where and how we position our sensor is very important. So we'll typically see our hyperspectral sensors used in any combination of the 4 methods shown here. And while there are nuanced pros and cons to each of these methods, you are essentially balancing the overall coverage of your imaging, measured in total area, with the desired resolution of that image, measured in area per pixel. So in this webinar, Carson's going to highlight a specific application in which we use the drone to provide the right amount of detail and flexibility for our mission. However, beyond just our hyperspectral data, we also always rely on ground truth data to compare our spectra, so we are confident in the classifications we're making. For this project, our ground, or our water truth data, came from our partners at Coastal Ocean Vision. To tell you all about that work, I'd like to introduce you next to the president of Coastal Ocean Vision, Scott Gallager. Scott?
Thank you, John. Can we go to the next slide, please? There we go. I'm Scott Gallager. I was a scientist at the Woods Hole Oceanographic Institution for 45 years, and we spun off a small company, Coastal Ocean Vision, with 8 to 10 of the engineers and scientists from Woods Hole. And we've been working together for many years, and so we formed a really nice team together. And we also have Fred Richards, who is our CEO, and has lots of small business experience. That's what really keeps this company going. Next slide, please. So our vision is to create tools to protect a variety of different aquatic environments, such as the ocean, concerning aquaculture for public beach activities, and drinking water, certainly, related to cyanobacterial HAB blooms, and commercial fisheries. And the intent is to use optics, optonics, and chemical sensors to detect these contamination sources before they become a public threat. So this is really the intent of where the company is and where we're going. If we could go to the next slide, please. We'll take a look at some of the tools that we have developed and are developing currently. On the top is a in-water plankton microscope called CPIX, and this images plankton of all kinds, from tens of microns to tens of centimeters. And it produces a dark field image, and it has onboard deep learning classification, so you can train it to identify any number of different types of planktonic organisms and particles. The next one down is a AUV, a Remus 600, but on the nose is a down-looking stereo camera. And it takes stereo images at about 6 frames per second and mosaics them together. And again, uses deep learning to classify targets and substrate on the fly. It can also detect man-made objects and integrate that with the side scan information on a much larger scale. The third product down is what we're going to be talking about today, and this is called HABstats, for Harmful Algal Bloom Statistics. It's a Raman imaging flow cytometer. So you can think of a flow cytometer as a particle imaging system that typically uses fluorescence to both detect the particle and determine pigment content. Raman imaging means that we're using Raman spectroscopy, which is a vibrational spectroscopy, much like FTIR, but is complementary to that type of vibrational spectroscopy. So as a cell goes by, the window, the imaging window, not only does a visual light image is taken of that particle, but also a laser, in this case 532 nanometers, is triggered, and a Raman spectrum is collected from that same particle. So we can merge spectral data directly with individual particles. And that's why this instrument is so important for purposes such as ground truthing, the hyperspectral cameras that we'll be talking about today. And then on the bottom is a full-blown seafloor observing system we call Ocean Cube. We've installed these all over the world and are implementing a number of our sensors, including HABstats, on these Ocean Cube observing systems. Thanks, go ahead. And finally, the CPIX, could you just go back to that last slide, please? The CPIX dark field imaging system We just had a couple of example planktonic organisms. And what's really key about it is that the dark field image is very clean and crisp, even though the current speed might be going very fast by the imaging window because of the high speed strobe system that we use. Okay, yeah, there we go. Okay, so yeah, some examples of copepods, fish larvae, radiolarians, and anything within the size range of 10 microns to several centimeters, we can get very clean images. And then, as I mentioned, we have deep learning classification on board that allows you to identify these targets after they've been trained. Okay, thanks.
My name is Carson Roberts from Headwall Photonics, and I'll be talking today about a preliminary study we have done of harmful algal blooms in Santuit Pond on Cape Cod on the east coast of North America. So, talking about harmful algae blooms, I want to first describe what the problem is. And so there are elements necessary to life. If you want to make a potato or a person, the main components you need are carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. Carbon is important. It's the largest component of most living things. It combines with oxygen and hydrogen to create carbohydrates like sugars and starches and cellulose, and hydrocarbons. Hydrogen and oxygen, they're in hydrocarbons and carbohydrates as above. And they're also in dihydrogen monoxide, which is in all cells and in all bodies of water, essential to life. Nitrogen is abundant in the atmosphere as the N2 molecule, but that molecule is very tightly bound and isn't biologically available. And it has to be broken down generally into some nitrate form like ammonia. And then it is available for living cells to make amino acids, proteins, and build the rest, many parts of cells. And so nitrogen is very often the limiting factor in the amount of biomass that can grow in any particular environment. That's one of the reasons that nitrates are very important as fertilizers. Sulfur is also essential to life, but it's generally not a limiting reagent in the growth of a particular species. Phosphorus is also essential to all cells. All cells have a cell membrane, which is a phospholipid bilayer. And if you see this picture of all these little cell membranes, every single molecule, every single little ball and stick in that has at least one phosphorus atom in it. And so nitrogen and phosphorus are limiting reagents. An excess of nitrogen and phosphorus in an ecosystem can lead to blooms of otherwise less abundant organisms. So the problem particularly for the harmful algal blooms, is human and agricultural waste. Now, human waste, because it takes nitrogen and phosphorus to build people, we excrete them in our waste, and the human waste, if it gets into bodies of water, can increase the amount of biologically available nitrogen and phosphorus, and can lead to blooms of otherwise less abundant organisms. Agricultural fertilizers. Again, you're trying to build cells, and so most agricultural fertilizers contain nitrogen, phosphorus, and potassium. So sewage, septic systems, those can put a lot of nitrogen into the atmosphere, because although all the solid waste can be collected in a septic system, the nitrates and phosphates are highly soluble, and they will seep through the groundwater and get into bodies of water. Runoff from over-fertilized fields can also increase the amount of nitrogen and phosphorus. And so, combined with the fact that we've got global warming, the temperatures of bodies of water are increasing. Most things are going to grow faster when it's warmer. That, combined with the runoff from human waste and from agriculture, can lead to harmful algal blooms, where some species that are toxic can grow out of control and make an entire body of water toxic. And at the bottom, I have two pictures of our study area. Now, these are data that were taken actually with our hyperspectral systems. It's RGB images from the hyperspectral systems. On the left, you can see the pond in November. On the right, you can see the pond in August. It's hard to see in November where our hyperspectral data are because they kind of match the color of the Google Maps. But you can see in August that the pond had turned very, very green. That is the harmful algal bloom, and that is what we are trying to measure to see if we can detect and quantify these with hyperspectral imaging. Now, describe the problem that leads to these toxic algae blooms, which is too much nitrogen and phosphorus coming into an ecosystem. Now I'll talk about the culprit. They're called cyanobacteria. They're also called blue-green algae. They're not actually bacteria. They're not actually algae. They're very primitive life form. There's a lot of evidence that cyanobacteria are the very first organisms to discover photosynthesis. All plants are green because they have subcellular structures called chloroplasts, which genetic analysis indicates are very likely descended from cyanobacteria. And when cyanobacteria first evolved, they led to a significant change in the atmosphere of the Earth. Prior to the development of photosynthesis, the Earth's atmosphere was reducing. It didn't have much oxygen, had no free oxygen in it. After the development of photosynthesis, the atmosphere changed to an oxidizing atmosphere because it's got oxygen in it. And as all of us know, oxygen is essential to the life of all animals and some plants. And one particular species of cyanobacteria called Chlorococcus is by biomass the most plentiful species on Earth. It lives in the ocean, and there is some evidence that it contributes to about 20% of the oxygen that we have in our atmosphere today. There are many, many different species of blue-green algae or cyanobacteria, and Dolichospermum, Veronicea, and Microcystis all produce toxins that can harm people and animals. And that's what we call the harmful algae in harmful algal blooms. So what is Headwall trying to contribute here? Well, the cyanobacteria, because they're also that are blue-green algae, have different photosynthetic pigments than normal algae. That's why they're called blue-green. And if it's a different pigment, it has a different spectrum. And with hyperspectral imaging, we can look at the spectra of pixels in our images, and potentially detect, oh, this particular spectrum seems to be like the harmful algae, the cyanobacteria, and maybe we can identify them. And the reason this could be useful is these blooms happen slowly. They're not there all the time. I'll show you a little bit about how in the pond that we've been studying, the population of the cyanobacteria rises and falls over the course of the year. And early detection can lead to early mitigation. There are some techniques that people can use to actually reduce the amount of cyanobacteria in a particular ecosystem after a bloom has started. And so what we've decided to do was to work with our partners at Coastal Ocean Vision to get what we call the ground truth. In this case, it's more the pond truth. And they're going to be going through and actually measuring the water at the same time that we are looking from the air with our hyperspectral imagers to potentially come up with a way that we can fly one of our systems over a pond or any body of water and say, yes, there are harmful algae here. No, there are not. Here's where they are. Here's where you should do your mitigation methods. Or this particular beach should be closed because we've detected too much of this dangerous material. So I'll give a little bit of overview of what the Headwall technology is that we're using to do this hyperspectral imaging. And the imagers we use are a slit scan imager. So on the left, I have a picture actually taken from some airborne data. You can see some striping in it. The aircraft goes back and forth and slowly builds up the image. It builds up the image line by line. So instead of taking the full picture, we'll only take one line of the image, and then we take another frame, which is one more line, and ideally we have some motion that allows us to slowly build up the image. Now, why would we want to take just one line at a time? Next. The idea is that if we take that one line of the image, so that's a row of pixels, something like 640 to 1,000 pixels across our detector, we disperse that so that each one of those image pixels is re-imaged at a different wavelength so that each pixel then has a spectrum. And so we take this single slit and we use the entire detector to get this full spectrum from every slit. And then we build up our image that way. And then we build up this hyperspectral image where each pixel has a spectrum. So how does the imager work? What we do is we have a 4-optic that makes an image onto a slit. Again, that's this first focal plane that's mostly dark, but transmits light just over a single row that's about the same size as one of our pixels. It is re-imaged onto a electronic focal plane array with a 3-mirror relay. One of those mirrors is a diffraction grating. And so because the diffraction grating reflects different wavelengths of light at different angles, we end up with the image I showed you in the last screen, which is one line of pixels re-imaged at many different wavelengths, typically on the order of 300 different wavelengths. And so each time we take a frame, we get, in the case of the imager that I'll be talking about today, 640 spatial pixels and 270 spectral pixels. And then we take another line, we move forward, take another line, we move forward, and we build our image up that way. So in addition to the hyperspectral imager, the research we did also, our payload included a LiDAR. LiDAR, just like radar, and it's light detecting and ranging. The instrument has 16 paired photodetectors and lasers. Each laser fires, and then the time to a return detected by the photodetector is measured. Because light travels at a constant speed, if you measure the time it takes for the light to get there and back, you know twice the distance to it. And then the lasers are spinning around. We're taking thousands and thousands and thousands of these per second. And then that gives us these points in space. And each point has a latitude, a longitude, and altitude, and some other metadata, including, and this is important for this research, the GPS time at which that point was collected. So on the left, you can see a picture of the drone that we used for some of this research. It's a DJI M300 with the Headwall Nano Hyperspec and a LiDAR on it. And it flew back and forth over the pond. And then in the right picture, you can see a small boat that has the detector that is actually sampling the water and measuring and counting the number of various species of plankton in it. And you should be able to see that pond is green. This was in August, and this was when the pond really was toxic. Fishing was banned, swimming was banned, and there was a tremendous amount of the cyanobacteria in the pond. So next, I want to talk a little bit about what our colleagues are doing in the boat. So they have a Raman imaging flow cytometer. And so the flow cytometer, cyto, I believe, means a cell, so it's a measuring cells. It's sucking up water. It's running it through a microfluidic channel, and then it gets into a little measurement chamber. And if there's a cell detected, a laser blows onto it, and it gets a Raman spectrum from that. And the Raman spectrum, similar to the reflectance spectra that we're getting from sunlight, could give you a very accurate measurement of what the pigments are, and that can be used to distinguish the various different species. They're using some sophisticated computers to figure all this out. And the net result is the folks from Coastal Ocean Vision are going back and forth over the pond in a boat. They are measuring the relative concentrations of all the different plankton species, in particular the harmful algae, at the same time that we are flying over them. Next, please. And so this picture, if you see in the left-hand slide, there's something that looks like a aluminum tube, because it's an aluminum tube. And inside of it is another version of this Raman imaging flow cytometer. And in the boat there, you can see our colleagues. They went back and forth over the pond. On the first image on the left, you can see before the flow cytometer is fully deployed. On the right-hand side, you can see it drags along in the water, and it's constantly sampling the water and geotagging and time tagging all the measurements that they're getting of the relative abundance of the various different plankton species. So here's another picture. We were out there in August the toxic algae blooms tend to be correlated with large rain events because that increases the runoff, and that in turn increases the biologically available nitrogen and phosphorus that can lead to blooms of these toxic algae, including Dolichospermum. And you can see this, a picture, they seem to form little rings. The cells are quite small, but there's enough of them that they really, really color the water. So in addition to this HABstats system that they're dragging around in a boat, there are 4 or 5 buoys that sit in the pond, and they measure the relative abundance, in particular, of the Dolichospermum harmful algae as a function of time. And I've got 3 red arrows there. We did our first flight over the pond, along with our colleagues in the boat, in June. We did another mission in mid-August, and we did one more mission in November. And you can see from the blue lines in the upper left-hand side that there were very few of the toxic algae in June. There were few also in November, and our mission in in August corresponded with near the peak of these toxic algae. And then one of the things that seems to affect the population of these harmful algae is chytrid fungi, which my understanding is they latch onto those little rings of the Dolichospermum and cause them to sink to the bottom and die. And there was a bloom of these chytrids, which led to a crash in the population of the toxic algae soon after our mission in August and well before our mission in November. So I'm jumping a little bit ahead here, but this is a map where I have mapped 3 different types of toxic algae based on their reflectance spectra, and I've circled several locations of the boat. So we're flying over the boat, and we can actually find the boat in the imagery. Because we have spectral imagery, we can take the image and we can classify each pixel according to what its spectrum is most like. And so I've built a library with several different kinds of trees, a couple of species of Elodea, which is a non-algae plant that grows in the pond, 3 different types of toxic algae, and 3 different spectra that I was able to associate with the boat in the imagery. So the next slide shows some of these images. So RGB images taken from our ortho-rectified mosaics of the hyperspectral images. And then below that, classified images. It's all pink because they're going through a soup of the toxic algae. And then that's how I can identify, oh yes, there's the boat there because I can see the fiberglass and the canopy and the white froth from its wake. And since I have the LiDAR, the LiDAR measures altitude. Well, the boat is on top of the water. The water is flat. And so anything that sticks up above the water can be seen in the LiDAR point cloud. All of these little things that look like little worms in our images. On top of, you can see sort of a grid pattern where we went back and forth, north and south and north and south and north and south. And you can see there the path of the aircraft. And it took us a little while to coordinate well with the boats, but by the end of our mission, we got 4 different images where the boat was very clear, and I can get the exact GPS coordinates and the GPS times for each of those boat locations, and we can use those to very carefully correlate the data that were taken by the boat at those exact times. Next. And so here then are the times and the GPS locations for them. This particular image, instead of being colored by altitude, is colored by GPS time, with blue being the beginning and red being the end. And so that's how I can extract from the LiDAR the actual exact locations and times of the boats for correlating these measurements. Okay, so here is a part of the study area where I've overlaid it on the Google Maps. The Google Maps, I don't know when these images were taken, but not during a toxic algae bloom because the pond doesn't show a lot of reflection. Water doesn't reflect a lot of light unless it's got a lot of stuff in it. and it had a lot of stuff in it in August. And you should also see that there's some lighter and darker areas here. That's because we're doing this, we're doing all these measurements where all our light is coming from the sun. And so if a cloud goes over, it gets a little darker. And if we get bright sun, we get a little brighter. And these intensity variations can have some effect on the classifications, but not a lot, because the method that we're using to do the spectral classification, which we call spectral angle mapping, is somewhat insensitive to the absolute intensity of the spectrum. Okay, so this is not images from the pond. This is some other work that we've done looking at in shortwave infrared spectra of geology. And this is an example of one way that we can do the spectral classification is to build up a reference library based on the ground truth. So in this case, this is out in Cooperite, Nevada, and we flew over this area, and our colleagues, the geologists, went out and collected a bunch of samples, and they brought those samples back to the lab. They used their geological techniques to identify what the mineral composition of each of their samples were. And then they took spectra of them from a lab spectrometer. They gave me the spectra, and then I used those as a reference library to compare to the spectrum in each of the pixels and say, okay, this spectrum, this pixel right here, I'm going to color it yellow because it's gypsum. I'm going to color it red because it's montmorillonite. And so that's one way of doing the classification. And when we continue this research, we will probably be doing this with some spectra taken from samples taken from the pond. But for this preliminary work, we did another method, which I'll try and outline in the next slide. And that is, I identify from the RGB image, I can say, this is very clearly a tree. This is very clearly the algae in the pond. And if you look towards the middle of the image, very near the shore, there's some little stripy areas where it seems much brighter than others. And so I chose 3 different classes of the harmful algae, 3 different classes of trees, and built up a spectral library, not from ground truth, but from pixels in the image. And so I chose a region that looked relatively uniform, got an average spectra from a few thousand pixels of that, built up the library, and then used that library to then classify all the pixels in the image. Okay, the next slide, please. And so here are a couple of images of the reference spectra. Anybody who's familiar with the spectral biology will recognize in the spectra of the trees what's called the red edge. And that's because chlorophyll, which is one of the main pigments in the blue-green algae and in trees, has a very strong absorption in the visible, but there's not enough energy in infrared light to drive photosynthesis. And so the chlorophyll pigment doesn't reflect past about 700 nanometers. And then you get the reflection of cellulose. And if you think of a piece of white paper, that's almost 100% cellulose. It's very reflective of light. And so you can see in the various trees, a lot of absorption, except in the green, around 550 nanometers. You could see a bump there. And then a high reflection out in the infrared. And then when we zoom in on these lower levels, you can see that there is significant difference. In fact, a strong bump I see around 650 nanometers that's much more associated with these toxic algae than it is with the trees or with the Elodea, which are the flowering plants that live in the pond as well. Next, please. So let's go. Again, we flew this pond 3 times, once in June before there was a lot of toxic algae. But if you look at our imagery from June, there's a few bits of pink there. So there was some, it just wasn't a huge bloom. And part of the reason that there wasn't a huge bloom, there was a lot of this Elodea. Now, Elodea is a plant that I was first familiar with from aquaria. It looks sort of like a feather brush, long strands. And in the pond, it made these large mats of it. In fact, when our colleagues were driving around in the boat, they kept getting stuck because there was so much of this Elodea. And one theory is the toxic algae bloom was late this June, and that may have been because there was so much of the LODA that it sucked up all of the available nitrogen and phosphorus, or a significant fraction of the available nitrogen and phosphorus, and delayed the toxic algae bloom. But based on our spectral classification, remember I've made these toxic algae various colors of pink, there is some, particularly around the shoreline, and it seems also associated with the edges of these LODA. So let's go to the next one. Now we have superimposed on our spectral classification data from the boat driving around. And we've got the Dolichospermum, the Microcystis, and then non-toxic green algae. And they're not getting a lot. These dots are associated with the concentration in number per milliliter. But there's some correlation of areas where we are seeing a little bit of pink in our images, and they're seeing some of these, some high concentrations of these toxic algae, but not yet a bloom. Okay. Now we go to August, where there was a lot of the toxic algae. And if you look at the RGB image, towards the center, I see a bit of a line heading off to the northeast. And those seemed different enough to me that I decided to make them as different spectral classes. And then we do the classification. It really looks like there is a differentiation. There's two clear spectral classes of the HAB in the pond. And if we compare that now with the data taken by the sampler in the boat, we see very clearly, again, on the eastern side of the pond, it looks as though the toxic algae is, the population is dominated by the Dolichospermum species. And on the on the west side of the pond, it's dominated by the Microcystis species. And this is, to me, the most intriguing result of the whole thing, is that it appears that we can distinguish between toxic algae species just by the spectral characteristics as measured by the drone flying over the pond. And so this is preliminary work, but this indicates that this is something that we will pursue next year with more studies and more ground truth. But again, it really does look like we've proven out the idea that we can see the toxic algae, and we can identify different species just by their spectral reflectance. And to complete the study, let's look at the data from November. So after November, remember I said there was those fungi that seemed to have caused a crash in the population. We fly over, again, the pond's not green, but our data seem to indicate that there are still some toxic algae. And most intriguing to me is in the northeast corner, in the top of this, we see a small area that's quite bright pink. That happens to be where a stream that is flowing through a cranberry bog, which is fertilized, is emptying into the pond and leading to more of, a small bloom of this toxic algae. And if we again look at the correspondence, again, up there where I see that little pink thing, big red ball, but the rest of the pond is mostly populated by the green algae. And so that's our basic results. Very encouraging. We will be working next year with our colleagues at Coastal Ocean Vision to try and extend this research. And I would like to acknowledge our main pilot is my colleague Charles Kepler. Scott Gallager from Coastal Ocean Vision ran the boat, did all the work. My colleague from our marketing team, Ross Nakatsuji, organized everything for us. And the rest of the people at Headwall, the people who build the imagers, the people who do all our work. And there were several of our colleagues who were with us. These missions generally take a bunch of people to get the drone in the air and get everything going. And thank you very much for listening to this talk.
Okay, and now we'd like to open it up to questions. We got a couple during the presentation, so I'll start with those. First, can this technology be used to ID small plants and trees? Is there a reliable source that provides individual species reflectance signatures, or will models need to be trained each time? Carson.
So the answer is, of course, is yes, it can be used to identify various different types of plants by their spectral signatures. I'm not aware of any particular library of these spectra, but we have certainly found that if we get spectra recorded with external measurements, we can put them into our models and do a good classification of the hyperspectral data with that. And then in terms of would it have to be trained each time, if you're looking at a particular place, you could go over at one time, build a library of the species that are in that particular area, and then all subsequent measurements, you could use that same spectral library.
Great. Is your technology available to lease or purchase? Mikhail?
Yes, so we have obviously systems that are available to purchase. If you would like some more information, you could contact us directly after this webinar, or feel free to call us. I think our contact information was listed. If not, just call our main number and we'll be happy to talk to you in more details about this question.
Did you perform any atmospheric correction over the Headwall data to convert to reflectance and account for atmospheric effects? Carson?
So yeah, that's a standard part of our data processing procedure. So our detector is silicon focal plane array. It produces data in raw digital numbers. We do a conversion to radiance using calibration that we do at our shop here. And then every flight we fly over a calibration tarp. We look at the radiance image, the radiance spectra from that calibration tarp. We have stored the reflectance spectrum of the calibration tarp, and we can then use that spectrum that we measure from the tarp to convert all the rest of our data from radiance to reflectance, and that has implicit in it the atmospheric correction for the day that we flew. We generally like to redo our atmospheric correction, that is flying over the tarp, about once every hour.
Did you find any relationship between pigments and the hyperspectral bands, Carson?
I'm not sure about that. That's a sort of subtle question that we're hoping to address as we continue this research in the next year, where we'll be doing some much more careful measurements of pure cultures of the various species in which we're interested.
How confident are you in your spectral library? Could some spectra in the library represent 2 or more different species due to confounding inherent optical properties?
It's my assumption, I'm happy to have Scott chime in on this, but I would say that all of our spectra represents mixtures of multiple species. And the differentiation that we showed in the August data is that on the right, on the eastern side, there is the Diclospermum species dominate, whereas the Microcystis species dominate on the western side. But I would be very surprised if there wasn't a mixture all through. And again, this is the sort of thing that we hope to generate some reference spectra from pure cultures going forward.
Scott, did you want to chime in?
Sure, and that is exactly why we need to ground truth the hyperspectral data. You have to know what is actually there in order to calibrate it. And by looking at mixtures of different communities and what their spectral reflectance is relative to species composition and pigment intensity on a species-specific basis and a community-wide basis is really the only way we're going to be able to get forward and deconvolve these spectra into species components. And we can do that now with the Raman spectral information because we have a single cell analysis. So each colony or each cell is known in terms of its morphology from the image, and then its molecular composition from the Raman spectra. So we can deconvolve the Raman information, and then we're going to use that to deconvolve the reflectance information from the hyperspectral camera. That's all work that we're proposing to do this coming summer.
Did sunlight cause any radiometric errors, Carson?
Certainly. In fact, if you look at the slide that we have up here in the RGB image on the western side, you can see all the different stripes from the different flight lines. And it looks like on the third flight line, we had a cloud go over. And that made the decrease the sunlight relative to when we flew over the tarp. And so that's going to create a general decrease in the apparent reflectance of everything. But the spectral angle mapping technique that we've been using for doing the classification is fairly insensitive to the absolute magnitude of the spectra. And so anytime you're doing hyperspectral imaging, you're far better off with constant light sources. And any variations due to clouds can cause some artifacts which may or may not affect the data.
All right, what software are you using to perform the image analysis, Carson?
So the image analysis, all the image analysis and the spectral analysis is done using the Headwall HyperSpec 3, or I'm sorry, the Headwall Spectral View software. And then we used Cloud Compare to analyze the LiDAR data.
All right, how many field Raman spectra are compared with hyperspectral imaging data to make the classification algorithm? And how many control samples did you use, Carson and then Scott?
So in this particular study, all of the spectra that we used for the classification were taken entirely from the hyperspectral data. And so we showed some correlation with the data that Scott got with Raman, but all the spectral classes that were used to make these pink and green images came from the field, from the hyperspectral system.
Right, and the intent of having Raman as the ground truth is that we can collect individual cells, as I've been saying, and we can do that on an order of close to 50 to 100 cells per second. So we can run the flow cytometer at a fairly rapid rate while the boat is moving at about 2 knots. And eventually we're going to put HABstats on a autonomous surface vessel and literally track the drone automatically. That's going to be happening this summer. But by taking a high number of Raman spectra, we can average those if we're interested in a species average, and because we have the information from the imagery for morphology at the same time. What we actually do is take the Raman spectra for each cell and barcode the image directly, and then use that to go into our deep learning convolutional neural network, which takes the information from the image and the barcode simultaneously to get a classification to species and also whether or not there could be toxin present. We do get information on toxin from some of the spectral peaks, and we've been able to calibrate that, particularly for microcystin and several of its congeners, whether or not it's at a high concentration per cell, usually in femtograms of microcystin per cell, or if it's below detectable limits.
This question says, we have a large prawn farm and see the benefit of using such a system for early identification of toxic algae in the ponds. This would enable early mitigation measures. Would this system be suitable for this application? Carson and then John.
Well, I'm always really happy to sell another system, but based on our results so far, it looks like this might well be a good way to get some early detection. And as we looked at the data from June and from August, when the pond was not completely full, we did see some local concentrations of the cyanobacteria. And so I think indications are that this might well be useful for that application.
Yeah, just to add to that, as long as the early indications are positive, we're definitely willing to look into this. As far as next steps, Mikhail mentioned earlier, you can reach out to us online or call our main number, and we can have a further discussion. But early indications are that yes, this would be suitable for your application.
Did you say that you used the nano? Wouldn't the hyperspec choline HP VNIR sphere perform better? Are you planning to test this one as well in this particular application?
So we did 3 flights this past summer. We did one in June, one in August, and one in November. And the June flight we did with the choline system, and the visible near infrared spectrometer is identical to in the choline system is a hyperspec nano. We did look at the shortwave infrared data, and there was nothing really compelling to us. We got a much better classification in the visible near infrared, and so that's what we think is the best system to use going forward.
Yeah, and just to add to that, when it looks like all the interesting information is in the VNIR portion, being able to focus to just the nano lets us fly the M300, which is more lightweight, more nimble, high-performing drone. So that's definitely an advantage of focusing within the specific range. The one thing I will add is this test was originally done with the nano hyperspec, the original generation nano. Future tests, we do plan on using the nano HP, which is the latest iteration. So it adds to the spectral and spatial resolution of the original nano. So hopefully get even higher resolution on future flights.
And I'd just like to add to that, I see there was another question about the spatial resolution of our data. And for the nano flights that we did in August and November, our ground sampling distance was 6.8 centimeters. So each pixel represents just a little under 7 centimeters.
Okay. Does your drone keep the same heading yaw the entire flight, or does it switch during the transects? Wondering how you eliminate sun glint.
So all these flights were flown north-south, and so we flew due south, then did a turn while not taking data, and then came back in and started taking data again flying north. So we essentially had 2 different headings. And in terms of glint, we were just lucky that it wasn't a bright sunny day and the water wasn't that choppy. If you look back at the data from June, there was a lot of glint from the sun, and that's always going to be a problem with this sort of airborne imagery.
Thanks for the presentation. How much is hyperspectral resolution?
Okay, so that's the one that I saw, and I said it's about 6.8 centimeters, 6.8 centimeters spatial resolution, and the spectral resolution is about 2 nanometers.
Does your drone keep the same heading yaw the entire flight?
We just answered that.
Off the shelf.
Can other off-the-shelf remote sensing software besides Headwall be used to analyze the hyperspectral imagery to perform 3D scattergram, ISO class cluster building, cluster groups, unsupervised classification into user-decided number of groups to get a generalized LULC moment in time maps?
Absolutely. All of our data are stored in the NV format, which was originated by the producers of the NV processing software. And some of the work that I do, I use Python programs that I've written using Spectral Python. We have MATLAB libraries that will open it. Our data can be analyzed in NV. It's basically standard hyperspectral data. And so any place that you've got analysis software, that should work just fine on our software.
Are there any irradiance sensors on board the UAV? Are the reflectance panels adequate?
We've worked a lot at putting an irradiance sensor onto the UAV, and we've just not found them to be reliable. The main problem that we found is that the apparent downwelling radiance will change as the drone pitches and yaws to and away from the sun. And so we've gotten all these results are with the reflectance panels. That is our standard technique so far. I will say that some of the people that use our systems will use our calibration to do the conversion to radiance and then use one of the atmospheric correction modules in ENVI, for example, to do their conversion to reflectance, and they're happy with the data that they get that way.
All right, one more. How automated is the HABstat system? What is it like operating them? Scott?
Yeah, HabStats is fully automated in the sense that you have to manually train it. If you're going to do automated classification, you use at least 100 to 200 images on a per species basis, and you set up a class, and then you build the deep learning model, and all that can be done remotely. So let's say this instrument is off in some pond or lake or river or the ocean, and it telemeters its data back to home base. The user can then build the various classes and telemeter the model back to the instrument, where it can immediately start using that in real time to analyze the water. So it's hands-on to a certain extent until you're ready to do automated classification, and then you push the button and it just goes off and it does it for months on end. And that's another good point, is when it's in the water sampling continuously, it does have anti-fouling capability so that the flow cell is kept clean for months on order. And we've had annual cleanup and repositioning of instruments over the past few years. So it's a long-term, highly automated system.
Fantastic, thank you, and thanks to everybody for asking questions. Just want to let you know about our next event, which is the CHRSE workshop, and that's an in-person workshop at the Center for Hyperspectral Remote Sensing Europe. That's April 19th and 20th at GeoConcept in Adelsschlag, Germany. CHRSE was formed in 2019 by Headwall and GeoConcept to support hyperspectral imaging technology, and it focuses on agriculture, mining, environmental monitoring, and infrastructure inspection applications. So that's going to be great. Please sign up. And thank you so much for attending. Special thanks to our panelists, Mikhail Jobran, John Margeson, Carson Roberts, and Scott Gallager. Thank you so much and have a great day.
Thanks, everybody.